Hello friends, welcome to this channel Science Place. I am Dr. Vandita Rathod. Today we are going to see how to prepare the sample for light microscope. This is the second part of the lecture, that is the study of microbial structure. So let's begin. In this lecture we are going to see how to prepare sample for light microscopy. This can be done by two methods, either wet mount or by making the smear and then staining the sample. So let's see what is wet and then staining can be either simple, it can be differential. Differential staining, when you are able to differentiate between the organisms, like by using gram stain you can be uh, group the organism into parts. It may be gram positive or gram negative, and using acid fast staining you can differentiate the organism, either it is acid fast or not. Apart from these two staining, there may be some special staining methods like negative staining, flagellar staining, and endoscope staining. So in wet mount preparation, it's a very basic method of to view the microorganisms, in which you just put a little wet part either of some culture, that is liquid culture. You can put a drop of liquid culture on the slide. This is a slide. You can put a sterile slide, and you can put some water or normal saline or iodine or some KOH, and then put your sample using a inoculating loop, mix it properly, and then cover with it a cover slip, and then you can directly see under the microscope. So it's a basic method and very useful to see the especially you can see the protozoans. To see the protozoans, it is very useful technique. And if you want to see that some organism is motile, moving something, so you can use this technique, and this is very helpful to visualize the fungus. The sample you can get from the fungal infection samples, you can have some skin, nail, hair, etc., that can have some fungal infection. So for fungal infection, potassium hydroxide has been used, because it's very alkaline in nature, that degrades the unwanted material that hampers in the vision of sample under the microscope. And you can also easily see the eggs and cysts and oocytes in the sample. So this is very basic technique of wet mounting, and after that we are going to see you can see the wet mount of a fungus. You can see the budding yeast is there, and single yeast is also there. The next is simple smear preparation. It's a very very basic method. You just need the glass slide, clean it properly, and spread the normal saline or some water if your culture is on plate. And if your culture is liquid, you can put directly few drops of liquid culture and spread with the help of loop, and then let it air dry. And after that, flame this smear by passing through over this flame at least 4-5 times, so that the organisms get fixed on the slide, it not moves on staining, it not run away on staining. And in addition to that, bacteria get died in this procedure, so it is a safe procedure. So once you make the smear, next is how to see this smear. To see the smear, you need a contrast. For this, you can use simple staining or differential staining, or uh, your some special staining technique that depends on the microorganism you want to see and you are suspecting in some sample. Suppose you are suspecting Mycobacterium tuberculosis, then you will not go for simple staining, you will go for acid fast staining. But if you are suspecting some Staphylococcus aureus, you will go for differential staining. Or just you want to see either some sample is having bacterial cells or not, or you want to see just shape of bacteria, then you can go for simple staining. Simple staining is simple because in this staining you use only single stain. Simple staining में आप you use only single stain to create contrast between the bacteria and the background. That's why it is simple staining. And usually commonly used dyes are the crystal violet, carbal fuchsia or methylene blue. So these usually take 5 to 10 second, like 5 to 10 second carbal fusion, 20 to 30 second crystal violet, and 1 minute for methylene blue. So once you prepare your smear, you can simply put on your stain on the slide, let it be there for the time as required for particular dye, and then wash it out, and then let it air dry and see under the microscope. This is the simple staining. Okay, the next is gram staining. Gram staining is really very famous and it's a very beautiful technique. Gram staining was first discovered by Hans Christian Gram in 1882, and he published his finding in 1884. He was a Danish bacteriologist, and this is a differential staining technique because you are able to differentiate bacteria into groups, either gram positive or gram negative. A gram positive bacteria appears violet in color because it is having a thick layer of peptidoglycan, and that is able to retain crystal violet dye, and gram negative bacteria are having thinner peptidoglycan layer or wall that uh, cannot retain the crystal violet. That's why it retains the counter stain, it captures the counter stain and appears pink in color, pink to red in color. So these are the simple steps of staining. Like first we use the primary stain, then mordant, decolorization and counter stain. So primary staining, in generally we use crystal violet. You can see here this is a fixed, these are the fixed bacteria on the slide. Once you stain these bacteria with crystal violet is a bluish to violet color dye. All bacteria take this stain and becomes blue to violet in color. This is known as primary staining because we are using only one stain in this procedure. Next, we use a mordant, that is a iodine and potassium iodide solution. This grams iodine solution, when we flood the smear with this gram iodine solution, it get inside the cell and crystal violet binds with this iodine solution and it fixes the stain inside the cell. Means now bacteria will able to hold it properly, it will not wash away in case of only gram positive bacteria. The next step is decolorization. In decolorization, that can be done by acetone ethanol solution or with 95% ethanol. On decolorization, gram stain bacteria captures the gram stain while gram negative bacteria loses its stain. And then on counter staining, that may be basic fuchsin or safranin, whatever the counter stain you are going to use, it will, the gram negative organism will capture the counter stain and appear, if you are using safranin, it will appear like red to pink in color. And there is no effect of counter stain on the gram positive organism. So basic idea is that after primary staining and using mordant, decolorization and counter stain, you are able to differentiate the bacteria either blue or pink, either gram positive or gram negative. But how it happens, what is the principle behind this? Let's see this. So first of all, this is, I mean, don't get afraid with this complex figure. This is really simple picture. Let's see how it is. Uh, Suppose this is the inner part of bacteria, and we are moving from inner to outer side. Okay. So first of all, the bacteria will having cytoplasmic membrane. This is this is the this is the cytoplasm, and then This is having cytoplasmic membrane. This second is cytoplasmic membrane. This cytoplasmic membrane is surrounded by peptidoglycan layer. Murine is also known as peptidoglycan. Peptidoglycan layer in one type of bacteria. However, in second group, this peptidoglycan layer is thin. You can see here, this is a thicker murine. This is a thin layer of murine. Okay, this is pink, this is purple. So, in gram-positive bacteria, there is a thick layer of peptidoglycan. However, in gram-negative bacteria, there is thin layer of peptidoglycan. And in gram-positive, after peptidoglycan, there is nothing. And peptidoglycan is also having teichoic acid some structures. But in case of gram-negative bacteria, there is also one outer membrane. Okay, so this is having two membranes, and in between there is peptidoglycan ka very thin layer. So this peptidoglycan is sandwiched between two membranes. This is cytoplasmic membrane, inner side, outer side, outer membrane, and in between there is peptidoglycan. Let it be a little simple. Forget all about the inner part. This is the outermost teichoic acid, murine or peptidoglycan layer, thick in nature. This is thin peptidoglycan layer surrounded by outer membrane. Okay, let it be more simple. I will remove teichoic acid and other things also. So this is the basic difference between gram positive and gram negative bacteria. Gram positive is having thick peptidoglycan layer. Gram negative is having thin peptidoglycan layer surrounded by outer membrane. This is the major difference. So how this difference is going to play the role in staining? Let's see. Principle of gram staining. What happens when you fix the bacterial smear? If this is gram positive bacteria, so it is having thick positive thick peptidoglycan layer, and if this is gram negative bacteria, it is having thin peptidoglycan layer and one outer membrane. Is it okay? I hope it is okay. Okay, and now you are putting crystal violet. What will happen? Crystal violet will go and stain the organisms. Both type of cells will be blue in color. Crystal violet will go inside the cells. Okay. Now what you are doing? You are adding grams iodine. Grams iodine will go inside the cell and it will bind with crystal violet. You can see here this blue color is crystal violet and this brown is iodine. So now crystal violet and iodine are going to bind and make a bigger complex that is known as crystal violet iodine complex, in brief CVI complex. See, this is bigger in size in comparison to crystal violet alone. है ना? Next is when you are going to decolorize, what is going to happen? The outer membrane of gram negative bacteria is going to disappear, going to dissolve. This outer membrane is going to dissolve in presence of decolorizing agent. And in addition to this, this is having thin layer of peptidoglycan. So this membrane is not having, this layer is not having the potential to retain the crystal violet iodine complex. So on washing, this is going to wash away. But gram positive is having a very thick peptidoglycan layer. So it will retain the CVI complex. Now you are going to add counter stain. Counter stain will reach in both cells. Counter stain will go inside both cells because it is smaller in size, but because it is always already having blue color, strong blue color, so you will not able to see the change in blue color. But here, because there is no blue color, this bacterial cell will appear as pink to red in color. I hope the principle is clear to you. You can have some words also for this principle, like cells are stained with crystal violet dye, then added with grams iodine solution. They form a complex of crystal violet and iodine inside the cell. This is larger complex molecule than from the original crystal violet and iodine solution. Then decolorizer like alcohol or acetone, they dehydrates the peptidoglycan layer, and shrinking and tightening causes the running away of large crystal violet from the gram negative cell, but gram positive cell retain this because of difference in peptidoglycan layer thickness. So after losing the crystal violet iodine complex, counter stain reaches inside the gram negative cell, and gram negative cell retains the lighter color of crystal violet. You can have the screenshot of this principle to write down in your exams. After that, let's see how to do this gram staining. You need a Bunsen burner alcohol for cleaning the microscopic slide, a slide rack, and microscope to view the slide. And reagents, you need crystal violet, this is the primary stain, grams iodine, acetone, ethanol solution as decolorizer or 95% ethanol, and counter stain, basic fusion or safranin, and water to wash. Let's see. Once you prepare your sample, you make your smear. This is the smear. You need to flood the smear with crystal violet for 60 seconds. In this procedure, all cells will retain crystal violet. Here you can see. And after 1 minute, just wash it and add some iodine. On adding iodine, wash means not with the simple, just mild wash, just to remove the add-on stain. And after adding orange colored iodine, it fix the complex inside the cell of gram positive but not in gram negative. After washing, just add decolorizing agent and wash away, then again stain it with safranin for 45 seconds in this procedure, and then wash again to view the sample under microscope. So you will have this kind of, you can have this kind of image, either mixture of gram positive or gram negative cells, or pure cultures of gram positive or gram negative cells. Originally cells look like this under the microscope. This is a picture showing gram positive bacilli, and this is the picture showing gram negative bacilli. So this is all about the gram staining. Application of gram staining, it's a very beautiful technique and a very huge application in clinical microbiology as well as general microbiology. This gram staining is a fundamental to phenotypic characterization of bacteria, and you must learn about the gram positive and gram negative cells. There may be question, write down the 5 gram positive cocci, 5 gram negative cocci. So just learn these as gram positive cocci. Just learn the genus first. Staphylococcus. Staphylococcus is gram positive genus with gram positive cocci. So you can add Staphylococcus aureus, Staphylococcus epidermidis, then Streptococcus also. Streptococcus also gram positive cocci arranged in chain, and it can be Streptococcus pneumoniae, Streptococcus pyogenes, etc. In case of gram positive rods, you can learn Clostridium difficile, Clostridium botulinum, Clostridium tetani, and Bacillus anthracis, Corynebacterium, diphtheria. And in case of gram-negative cocci, this is very important. Gram-negative cocci, you can have Neisseria meningitis, Neisseria gonorrhoeae, Neisseria lactamica, Moxella bovis, and Moxella lactunata. Then gram-negative rods can have E. coli, Acinetobacter baumannii, Pseudomonas aeruginosa, Klebsiella pneumoniae, and Salmonella typhi. This is really important. These organisms are really important in terms of clinical microbiology. In case of infection, Acinetobacter baumannii is the most horrible organism right now. As per the list of WHO, it has been because resistant organisms are growing continuously. So carbapenem resistant Acinetobacter is on the first priority by WHO for discovery and development of new antibiotics. Okay, and apart from gram staining, you are not able to see all organisms in the by using gram staining. So like exceptions are like Mycobacterium tuberculosis, it needs acid fast stain, endospore, and to observe the thin structures you need different kind of staining. We will talk later about this. So this is about the gram staining method and application. Next we will discuss about the acid fast staining. Okay, so thank you for watching this video, and if you are liking this, please comment about the lecture so I can come to know if you're liking this or you need something add on. Thank you so much. Please like and subscribe this channel.